Welcome to Excellent Grades Academy. This is Dr. Byson EM. Today we're looking at the urinary system, which is Histology Lab Practical Lab number 19. Okay, let's get into what we're looking at today. The learning objectives are as follows: to identify the organs that make up this system from their microscopic appearance, to discuss the embryology and gross anatomy of the entire system, to describe the detailed features of the transitional epithelium, which is also known as urothelium, to discuss the functions of each organ, to distinguish the ureter from the gut tube, to give the common conditions affecting the organs in this system. All right, now the urinary system is a very unique system that is made up of the following organs. It's made up of 2 kidneys, 2 ureters, a bladder, and a urethra. Now, this schematic diagram here is the kidney. It is made up of the outermost structure known as the fibrous capsule. Underneath the capsule, we have the cortex. This part here is the cortex. And under the cortex, we have the renal medulla, which is made up of the renal column and the pyramid. The medulla will now form calyxes, minor calyx and a major calyx, which will drain the urine into this part here, structure known as the renal pelvis. The renal pelvis will connect the ureter to the kidney. Okay, now before we look at the histological features of this system, let's look at the functions of the kidneys. Number 1, to regulate the balance between water and electrolytes, excretion of metabolic wastes, excretion of many bioactive substances, for example, drugs. Most drugs are excreted through the kidneys, and regulation of arterial blood pressure by secretion of renin, which is a hormone that will cause a change in the diameter of blood vessels. This is a histological picture of the entire kidney. So the outermost structure here, this is the fibrous capsule. So this is the fibrous capsule here. Under the fibrous capsule, you can see this heavily bluish stained structure. That is the cortex. So let me just label it. This one here is the cortex. Under the cortex, you can see this structure here that is not as heavily stained as the cortex. This structure here is the medulla. The medulla will drain into the pelvis. So this structure here is the pelvis. The pelvis will now drain the urine into the ureter. So this one here is the ureter. So the part where the ureter will connect to the kidney is known as the hilum. So this part here is called the hilum. The hilum is also where the vessels will enter and exit the kidney from. The renal artery will enter the kidney at the hilum, and the renal vein will exit the kidney at the hilum. So you're supposed to draw this entire histological diagram of the kidney and label it the way I have shown it to you. Let's move on. Let's look at the kidney's cortex. This is the kidney cortex. Our focus on the kidney cortex is this structure here. Let me just draw around it. This structure here is known as the renal corpuscle. So it's the renal corpuscle. It's a functional unit of a kidney. This part, the outermost part of the renal corpuscle, is the Bowman's capsule. So this is the Bowman's capsule. And then what you see inside here, this is the glomerulus. Glomerulus. The space between the Bowman's capsule and the glomerulus, this whitish space around here, that is the Bowman's space. Bowman's space. So we can't really appreciate the structures inside of the renal corpuscle. Something that we're going to do when we look at the histological picture at high power. But what you see near the renal corpuscle, this that has nucleus here, which is simple cuboidal epithelia and has a lumen. This one here is the proximal convoluted tubule. So the proximal convoluted tubule. It is denoted as the PCT. So it has an irregular lumen and its walls are thick. But when you look at this has got an irregular lumen and its walls are thin. This is a distal convoluted tubule. Okay. So now let's look at this same structure at high power. This is the structure at high power. This part here in the middle, this is the glomerulus. And then this is the Bowman's capsule. The space in between the Bowman's capsule and the glomerulus, this is the Bowman's space. So the Bowman's space. Inside of the glomerulus, we have the mesangium. The mesangium are cells that have got contractile properties that act like smooth muscles. So they are mesangium that are inside of the glomerulus. They are called intraglomerular mesangium. And they are mesangium that are outside of the glomerulus. They are called extraglomerular mesangium. And those are part of the juxtaglomerular apparatus. Okay. So what you see here is a proximal convoluted tubule. It is even labeled PCT. So it is made up of simple cuboidal epithelia. These nucleus that you see, these are the nucleus of the cuboidal cells that make up the proximal convoluted tubule. And then this one here that is irregularly shaped, and its walls are not as thick as that of the PCT. This is the distal convoluted tubule. So this is the distal convoluted tubule. What you see here, this structure here, this is an arteriole. So this one here is an arteriole. So I expect you to draw this at high power and the low power that I showed you, focusing mainly on the renal corpuscle, laboring the glomerulus, the Bowman's capsule, Bowman's space, mesangium, the PCT, the DCT, and the arterioles. All right, let's look at the ureter. This is a human ureter at low power. The ureter, the function of the ureter is to pass urine from the kidneys to the bladder. Okay. So transmission of urine. Transmission of urine from kidney to the bladder. One feature of the ureter that you must know is that the ureter has got an irregular lumen. Irregular lumen. So as you can see, this lumen here is irregular. It's not regular at all. It is irregular. So if you follow it properly, it is an irregular lumen. The lumen is covered by transitional epithelium, which is also known as urothelium. Under the urothelium that rests on the basement membrane, there is the lamina propria. So this part here. The lamina propria is simply made of areolar connective tissue. Under the lamina propria, you've got the muscle layer, which is also known as the muscularis propria. The muscle layer of the ureter is made up of two muscles. So two muscle layers. You have the inner longitudinal muscles and the outer longitudinal muscles. Something that we're going to see when we see the high power representation of this histological slide. And then under the muscularis propria, we've got the adventitia. The adventitia is a structure where blood vessels are found. So here, this part here, this is an arteriole. How do you know that this is an arteriole? There's blood inside. And it has a thick lumen. And then these ones here, irregularly shaped. These are venues. They're irregularly shaped. And their lumens are small. The adventitia is also made up of adipose connective tissue, made up of a lot of fats, adipose connective tissue, and irregular connective tissue. So this one here, this is irregular connective tissue. You can see it, the way it's running. It's called collagen fibers that are irregularly arranged. It's also made up of nerve endings. All right, so now let us look at so this is the ureter at high power. So this is the ureter at high power. So you can see that the lumen is irregular there. Let me just move this. Okay, so this lumen here is irregular. So let's just label it irregular lumen. The lumen is covered by urothelia, which is transitional epithelia. And then under the transitional epithelia, you have the lamina propria, which is made up of areolar connective tissue. And then you have the muscularis propria, which is the muscle layer. So this is the inner longitudinal muscles. So inner longitudinal muscles. And then these are the outer circular muscles. Two muscle layers. And then under it here, you've got the areola. Rather, you have the adventitia. It is made up of adipose connective tissue. So adipose connective tissue. You can see this is fat cells. And then you've got the arteriole here. So this one here is an arteriole. And then these here, the irregularly shaped, these here are venues. So this is the ureter. Okay, so this is the ureter using another stain. So what we were using in the previous slide is hematoxylin and eosin. That's why it appeared like this. Here we are using Augean blue, so it appears blue. The function, this is the function transportation of urine. Okay. So this is the irregularly shaped lumen. And then under the lumen and under the urothelium, you've got the lamina propria. You can see here longitudinal, inner longitudinal muscles, outer circular muscles. And then you have the adventitia that is surrounding it. So this is simply the cross section of the urethra. Cross section. You can pick any urethra and draw. Just make sure you label everything properly. This is still the urethra that is labeled. Okay. Let's move on to the urinary bladder. The urinary bladder stores urine. This is the function. So it is made up of the urothelium. So this is a urothelium, the transitional epithelia. Transitional epithelium. Under the transitional epithelium, you've got the lamina propria. So this is the lamina propria here. Lamina propria. It is made up of areolar connective tissue. And then you've got the detrusor muscle. In the bladder, you have what you call the detrusor muscle. Detrusor muscle. The detrusor muscle has got 3 muscle layers. 3 muscle layers. This you must know. It has the inner longitudinal muscles. Okay. So the inner longitudinal muscles. Longitudinal muscles. And then it has the midsecular muscles. Midsecular muscles. And then we have the outer longitudinal muscles. Outer longitudinal muscles. And then after the muscularis layer, you've got the adventitia, which has got blood vessels and nerve endings. Let's look at another histological diagram. So this is a urinary bladder, and this histological diagram focuses on the epithelia of the bladder. So you notice that the epithelia of the bladder, the transitional epithelia of the bladder, has got 3 layers of cells. The uppermost layer of cells are called umbrella cells. Okay, so these are UM cells. These UM cells prevent urine from crossing the epithelium and entering the parenchyma of the bladder. And then you've got the immediate cells. These immediate cells are simply columnar cells. So you must know the immediate cells are columnar cells. And then you've got basal cells. The basal cells are cuboidal cells. They are simple cuboidal cells. So this is how the transitional epithelia of a bladder is formed. So you know that this epithelia rests on a basement membrane. So there's a basement membrane here where the epithelia lines. So this is a basement membrane. And then under the basement membrane, you've got the lamina propria, where you've got arterioles and venues. Here you can see in the arterioles and in the venues, the RBCs inside here. There are red blood cells in the inside. And then under the lamina propria, you've got the muscle layer. So this slide, you're going to draw and you're going to focus on the urothelium and the layers of the urothelium. 3 layers of the urothelium. Okay. So we've already looked at that. This slide is focusing on the muscle layer, the detrusor muscles, which has 3 layers of muscle. The inner longitudinal muscles, the midsecular muscles, and outer longitudinal muscles. So you've seen these, number 1, these are the inner longitudinal muscles, the midsecular muscles, and the outer longitudinal muscles. All these muscles are smooth muscles. They are involuntary. This is the same, just focusing on the detrusor muscle. You see, it's a very big muscle. Inner longitudinal, midsecular, outer longitudinal muscle. It's a very big muscle, the detrusor muscle that is found in the bladder. This histological diagram here is just labeled the layers of the muscles. The longitudinal muscles, their smooth muscle, middle circular muscle, and the outer longitudinal muscle. All right. And then we've already talked about the layers of the transitional epithelium in the bladder. Okay, so the common conditions in the urinary systems. Number one, cancers of the bladder. You can have renal cancer at any point. You can have bladder cancer. You can have cancer of the ureter, cancer of the kidneys. Incontinence. You're not able to control the flow of urine from your bladder. So that's incontinence. Kidney stones. Your stones can block your urethra. They can block your ureter most commonly, and you have problems. Kidney failure, where your kidney is failing to carry out ultrafiltration and formation of urine. So you won't be able to remove waste products from your body, and that will kill you. Urinary tract infections may affect your kidneys and may cause kidney failure. After that, you have this exercise. You do a well-labeled nephron, the type of epithelial lines that lines the renal papilla. List the components of the juxtaglomerular apparatus and their functions. List the components of the glomerular basement membrane. What podocytes are and their functions. What mesangial cells are and their functions. The type of epithelium that lines the ureters and the urinary bladder. We've already talked about this. And why this epithelia is unique to these organs. Transitional epithelia is unique to the urinary system. It's not found anywhere else. With diagrams, compare and contrast the histology of the ureter and the urethra. What are the characteristics of nephrotic syndrome? All right. With that, we've come to the end of this video. You can register with us for anatomy and all the courses in medicine at +260 975 497790. Learn with the best. Dr. Byson EM, Excellent Grades Academy, signing out. I'll see you in the next video.